Now, I told you this lecture is going to be a 2-star lecture, meaning that it's kind of complementary, right? However, due to recent events, due to some things that I'm going to explain, some concepts, we're going to upgrade it and give it a 3, even a, you know what, here, 3.5. There's another star, let's just cut it in half. So it's 3.5 lecture. Why? Because there are some important concepts I'm going to be talking about here. Now, the concepts, they're not directly related to the NAD biosynthesis. However, they are very important in other areas of biochemistry, let's see. Okay? Now, that being said, let's start with the NAD biosynthesis. Now, NAD biosynthesis, by the way, we can get NAD by 3 ways. Number 1 and 2 is from vitamin B3. Right? Now, vitamin B3 is, by the way, it's like an umbrella. It's called niacin. It's called the niacin, right? Now, vitamin B3 can come in 2 forms. It can either come in the nicotinic acid form, okay, here, nicotinic acid, or it can come in a form called nicotinamide, right? Nicotinamide. Now, most supplements are going to be in the form of nicotinamide. Rarely is it going to be nicotinic acid. You see, nicotinamide, first of all, the bioavailability of it is really good, right? Nicotinic acid, not so much. Listen, nicotinic acid, usually, mostly, we find it in plants, right? It has low bioavailability, right? So really the absorption rate, the utilization rate is kind of low. Nicotinamide, on the other hand, really we find it in meats, in liver, in poultry, in eggs, etc. Right? Now, when you buy a supplement, usually it's in this form. In fact, make sure it's in this form and not in this form. Nicotinic acid, oh, by the way, mostly they refer to nicotinic acid as niacin, and therefore in pharmacology, when you hear that we give niacin to really increase the HDL or decrease the LDL and the VLDL, is going to be in the nicotinic acid form, the niacin form. Nicotinamide, we call it, I think, niacin, it's niacinamide, I think, right? Niacinamide. Okay, so we're talking about biosynthesis. We're not going to dive into vitamin B3, just, for explanatory purposes only. So, a source could be, or we could start with nicotinic acid, we could start with nicotinamide. Okay, what's the third? The third is something called the de novo synthesis, meaning that I'm building it from scratch. So how can I get NAD plus from scratch? Well, that's something through an amino acid called tryptophan. Okay? Now, the tryptophan, we're not going to talk about it that much, because honestly, for all intents and purposes, the tryptophan or the de novo synthesis from tryptophan is not biologically important, and mostly for our purposes, it's not important at all, right? Now, there is a book by Stephen R. Covey, I think his name is. It's called The 7 Habits of Highly Effective People. Now, one of those habits, or one of the phrases that really stuck with me from the book, was always start with the end in mind. So I'm going to apply it to this lecture. What's the end? This is the end. This, ladies and gentlemen, remember, this we said is the nicotinamide, right, base. And this is adenosine, right? And so all of this structure here, we're going to call it the NAD+. So, whether we're starting from tryptophan, or nicotinic acid, or nicotinamide, the eventual base we want to reach, eventually, we want to reach this base, and eventually we want to reach this structure. Now, there are 3 pathways, again, I told you. If we're using nicotinic acid, it's going to be called the Preiss handler pathway, if we're starting with the nicotinic acid. If we're going to start with the nicotinamide, we call it the salvage pathway. Mostly because nicotinamide is recycled in the cell, however, it can be also in a dietary form. Still, we call it the salvage pathway. And the third one is the de novo pathway, right? The tryptophan. So let's start with the nicotinic acid. And believe me, listen, it's extremely simple, extremely easy. However, there are some basic principles that along the way, along this lecture, I want you to imprint in your brain just to get a basic idea about how things really go on in the body. This is going to be very important in the purine salvage, in the Novo synthesis of purines and pyrimidines, okay? So focus, okay. Let's start with the nicotinic acid. Now, I'm going to tell you a fascinating story here. One time, a group of scientists, they were bored. You know, scientists, sometimes they have nothing to do, it seems. And so, you know, one of them was smoking, and, you know, his friend looked at him and said, listen, let's oxidize your cigarette and see what we find. And that one was like, yes, please oxidize me. He said, no, I'm going to oxidize your cigarette, right? So what does oxidation mean? Oxidation is simply burning it, right? But you burn it under, let's say, some regulated or some controlled conditions. Anyway, not important. When they burned the cigarette and they looked at the chemical structure of it, right? Yeah, I know. You're probably getting bored. Chemical structures, again, this is very fascinating, believe me, right? So basically, what they found was a ring, which is a pyridine ring, okay? And that was attached to a pyridol ring, right? Okay, great, right? So what? Now, this is the nicotine. This is nicotine. Of course, it has some double bonds, right? Not important. Anyway, this is nicotine. So when they oxidized the nicotine and they burnt it and stuff, what happened? This ring completely disappeared, right? In its place was what? Was a carboxylic acid, right? Okay, that's very interesting. So they looked at it and said, yeah, you know what? Since that was nicotine, we're going to call this nicotinic acid. This is nicotinic acid. Why nicotinic acid? Well, it's derived from nicotine, and the acid part is this, the carboxylic acid. Now remember, we said in the, if you watch the lecture about the acid and bases in the biochemistry or the biochemical concept, we said that an acid is something when we have the pH of 7.4 and it donates a proton, we call it an acid. That's where the nicotinic acid name came from. Okay, good. In like the 1900s, like 1910, 1915, 1920, around that era, there was a big problem that happened in the United States. That problem was there were people that were coming in with symptoms, you know, the places where they were exposed to the sun, they were like inflamed, so we called it dermatitis, right? They had like frequent bowel movement, they had diarrhea. And some of them developed cognitive symptoms. So it was like they had brain fog, they couldn't recall events, etc. And this was widespread. Well, the scientists, the doctors later on discovered that this was pellagra, because those communities, generally speaking, were feeding on corn, right? Now, we're not gonna dig into the vitamin B3 deficiency and how it happens. That's lecture number 3. However, and so, you know, they tried to solve the question, why is this happening? Basically, they found out that it was vitamin B3 deficiency. So they took the vitamin B3, the niacin, right? And then they like put stuff on it and they really fragment it into its chemical or basic chemical structure. What did they find? They found nicotinic acid. Wait. So you're saying that if I take nicotine and I burn it, it gives me the same exact compound as vitamin B3. I'm telling you that's exactly what's going to happen. Of course, don't go and, you know, burn your cigarettes and like, you know, put the ashes in water and dissolve it and drink it. First of all, you're going to get a very small amount of nicotinic acid. Second of all, it's not going to be, most of it is not going to even be nicotinic acid because you're burning it in uncontrolled manner. Third of all, what the hell is wrong with you? Who drinks ashes? You're going to be drinking tar and like a lot of trace elements and like carbon monoxide, and you're going to poison yourself before you even get a trace of nicotinic acid. Not important. Okay, great. So now we have the nicotinic acid. How does it look? Well, it looks exactly like this. It's a pyridine ring. By the way, a pyridine ring simply means that there's 5 carbons, or it's a 6 carbon ring, right? So we have 5 carbons and 1 nitrogen. Not important. not important, okay? So this is nicotinic acid. How do we go from having nicotinic acid, which looks like this, right? Oh, listen, I want to tell you a story. Now, let me continue the story. So this is nicotinic acid, right? Anyway, and nicotinic acid is going to cure pellagra, right? And so Big Pharma comes in and they are like, yes, please, money. I love me, my money. And he goes to his scientists and says, listen, people, let's sell this. And maybe, you know, of course, the official story was, let's not call it nicotinic acid, let's call it niacin. Because we don't want people to, this is the official story, we don't want people really to associate the, you know, smoking, because it's nicotine, with something good, which is vitamin B3, a supplement. The real story was that Big Pharma was extremely happy. Now we can make a lot of niacin, or still the name wasn't niacin, we can make a lot of this compound, which is nicotinic acid, and we're going to make a lot of money. Now, the problem is, it's called nicotinic acid. I mean, who the hell is going to buy something, a supplement, that is, it's called nicotine, so you know, probably associated with cigarettes, which it is, and it has acid in it. No, 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 we can't do that. So the Big Pharma guy goes to his, you know, his team and stuff. He says, like, listen, I pay you good money. I want to sell this. Do something about it. Do something about the name. And so they were sitting down, right? And they were thinking, well, you know, it's a vitamin, right? So, and it's nicotinic. So Ni, you know, it's acid. Nia, it's a vitamin. So let's call it niamin. Woohoo, right? But you know, it's not really catchy, so instead of the M, let's put C. Now it's called niacin. And then they sold niacin. And then this dude became extremely happy because he became extremely rich. Okay? Now, let's remove these dollar boys, dollar eyes, and continue. I just like to tell these small stories. I think it makes me fun. Probably makes the lecture too long, too. Anyway, let's start from this and come here. Okay, so what is the first step that must happen to the nicotinic acid for us to take it from here and make, make this? Okay, first of all, let us attach a, we must make it into a nucleotide, right? So what do? Let's attach a nucleotide to it, or at least let's attach a sugar, right? That has a phosphate on it. Of course, by the way, when there's no base on the 1' carbon, it's a hydroxyl group that's going to be present. Okay, so here, this is a ribose sugar, right? This is a sugar, here's 4', here's 5'. It has a phosphate on it. The problem is that if you take this nicotinic acid and you want to place it directly on this, by the way, this is called ribose, because it's a ribose sugar. It's called a ribose 5, because on the 5' we have 5-phosphate, right? Because we have a 5-phosphate on the 5'. Okay. So we try to come and take the nicotinic acid, and by the way, this doesn't only go for like the nicotinic acid. No, it goes for adenine, guanine, hypoxanthine, whatever. You cannot go and simply attach a base, and nicotinic acid is a base here. You can't take it and attach it to the hydroxyl group. That doesn't work. You need energy. Okay. So what do? Well, first of all, let's add something that gives us energy. What's that something? Well, we have to add 2 phosphate groups. However, the 2 phosphate groups aren't going to be, you know, just running around in the cell. No, they're always attached to something. And usually that something is ATP. So what happens is you have here an adenosine triphosphate. Remember, we said it's the energy currency of the cell. And this is only one of its uses, right? And so you have an adenine here as the base. I'm not going to draw it, right? I trust you know that you're doing this in sequence, in order, you already know this. So you've got an adenine, right? Now, the basic concept is, or the basic idea is that we want to take these 2 phosphate groups and put them in, or replace the hydroxyl group with them, right? So, what's the enzyme that's going to do this? I mean, let's look at it. What am I going to do here? I am going to transfer 2 phosphates, and I'm going to put them on the ribose 5, right? So I'm going to do this, And so this is going to have 2 phosphates here. Now, if you watch the lecture on the phosphoribosyl pyrophosphate, you're going to know that this structure, when I add 2 P's, is going to be now called a right? Here's phosphoribosyl. And since these 2 phosphates, we call them PPI, or inorganic pyrophosphate, so they're pyrophosphate. So phosphoribosyl pyrophosphate. So the enzyme that does this is going to be called phosphoribosyl pyrophosphate, sorry, this is a P, phosphate synthetase, because, well, it's synthesizing it, right? Synthetase here. Okay, everything's nice. So I took 2 phosphate from here, I put it here, and now I have the this is no longer ribose 5-phosphate, it's now called phosphate ribosyl pyrophosphate, right? Good. And now what happens? Well, now the nicotinic acid comes here, right? And it's like, let's go and let's do this, and let's attach myself to this structure right here. By the way, when I put the 2 phosphate here, before I put them, it was a ribose 5-phosphate. It's kind of wimpy, it's kind of calm, it doesn't want to do anything really. When I put the 2 phosphate groups here, now we call it a loaded ribose sugar, or a loaded ribose, an active ribose. Now it's ready, to enter in a lot of pathways. This thing here can enter into the de novo synthesis of purines, of pyrimidines, right? Of nicotinic acid. So it's happy, it's loaded, and a lot of things. We're going to talk about them in the future. Or we already have a lot of lectures about some of them. Anyway, the nicotinic acid goes, and what happens here is it's going to attach, these 2 phosphates, by the way, are going to be used as energy now, right? And the nicotinic acid is going to be attached at the 1' to the 1', right? Okay, to be more specific, the base is attached at the 1' of the ribose to the 1' of the bases, right? Because the carboxyl group should be at carbon number 3, here's carbon number 2, and here's carbon number 1. Okay, great. Now, let us remove these, although we may, let's just remove them, don't worry. If we need it, we'll write it really quickly. So, what do we have so far? Well, we have a base, right? Which is the nucleic acid, right? It's attached to a sugar and attached to a phosphate. Of course, this sugar is no longer, a 5-phosphoribosyl pyrophosphate here. This whole structure is called nucleic acid mono. I mean, you would think phosphate, right? It's true. You could call it monophosphate. However, they call it nucleic acid mononucleotide because it's referring to NAD. That's all. Okay? Good. So, what's happening here? What did we do so far? Well, we have this structure, but on the 3rd carbon it's kind of weird. It shouldn't look like that. Okay, no worries. And now we have to add AMP, right? Adenosine monophosphate. So great. You're thinking now, let's take the carboxyl group and change it into an amide group, right? Not so fast. Before we can do that, listen. I mean, if we were in like a biochemistry lab, we could do that. It would be easy, right? You just, you take the hydroxyl group, because listen, COOH is nothing but a carbon double bonded to oxygen with a hydroxyl group. And so you're thinking to yourself, well, listen, all I have to do is remove the hydroxyl group here, right? And put an amino group. And if you're in a lab, yeah. But in a human body, we can't do that. Okay, so what do we do first? First of all, I have to attach this nucleic acid mononucleotide, I have to attach it to AMP, adenosine monophosphate. Okay, so let's do that, right? Let's do that. I'm gonna keep this because we want to see where we're going with all this. Let's make it blue. Okay? Now, the thing about okay, listen. This process is going to be called adenylation. And I'm going to tell you exactly what adenylation is. It's very easy. No. So let's look. The concept so far is the formation or taking the 5 or the ribose 5-phosphate and making it into a loaded sugar or an active form of ribose sugar, which we call the phosphoribosyl pyrophosphate, right? It's also, by the way, called the 5-phosphoribosyl 1-pyrophosphate, but that's not important. Okay, so the concepts we have learned so far is that the base is always going to attach to the 1' and it's going to be attached to the 1', right? There's number one. Number two, we told you and we said that we need to load the sugar so we make it active, right? And we showed you how we do that. Okay, and we talked about, of course, the PRPP synthesis or synthetase enzyme, synthase. Okay, now, what about the adenylation? What does adenylation mean? Adenylation, listen. Here's a phosphate, and here's a phosphate, and here's an adenine group. What do you call this? And this is a ribose sugar. We call this adenosine triphosphate, right? ATP. Great. What is the adenyl? Adenyl is this portion here. It's simply the portion of ATP that is AMP. Instead of calling it AMP, because it's part of the ATP, we call it adenyl. That's all it is. So adenyl is simply AMP, right? But because it's a portion of the ATP, we call it adenyl. That's all it is. Okay, great. So what happens here? Well, basically, there's an ATP, right? And there's an enzyme. And can you guess the name of that enzyme? That enzyme is going to be called nucleic acid mononucleotide adenylyl transferase, because it's going to transfer the adenylyl portion and stick it here. Okay? In fact, listen. Let's make it. I know it's going to take a little bit more time, but for all intents and purposes, it's extremely important. Here's the nitrogen base adenine, right? And here are 3 phosphate groups. Good. Now, the enzyme is going to take this portion, which is the AMP, which, because we took it from ATP, we call it adenylyl, and it's going to, literally, it throws these 2 phosphates. Again, these phosphates are going to be used energy to drive the reaction, okay? And it links the AMP portion, or the adenyl portion, it links it to what? It links it through an oxygen bond to this. So what do we have so far? I mean, sure, yeah, maybe it's flipped, the drawing, but basically here's the adenine, and here's the adenine again. Oh yeah, here, it's adenine. This is adenine, the nitrogen base. Here, for completeness sake, I guess. Don't want to confuse anyone. Right? And here's the NH2, the amino group, right? So here it is. And it's linked to what? To a phosphate, ribose sugar, nicotinic acid. Great. So it's almost so far. And again, by the way, this is a phosphodiester bridge, we call it. Why? Because there's phosphate number 1, phosphate number 2, and they're bridged with one another. Okay. And the enzyme, it's nicotinic acid mononucleotide adenyl transferase, right? Because we transferred the adenyl group. Okay. So what's so different about this? Well, this is exactly the same, right? Here it is. The ribose sugar is exactly the same. The phosphate exactly the same. Here's an oxygen group, here's an oxygen group, here's a phosphate, here's a phosphate, here's the ribose, and well, yeah, here's the ribose. Here's the, the 6-carbon ring, the pyridine ring, and yeah, you'd be right, here's the pyridine ring. But on the third one, here's number 1, 2, and 3. On the 3rd carbon, here there's a carboxylic group, and here there's going to be an amide group. Okay, so easy. Let's go and remove this carboxylic group, and remember we said that it's carboxylic, it's like this, right? So this carbon here is going to be like this. Let's take the hydroxyl now, and put an amino group, and then we're done. We have NAD. Yeah, you guessed it. No, it doesn't work that way. Listen, we have an enzyme, right? Of course we do. And this enzyme has 2 pockets. And before you say anything, it does not look like an ass. This is a very respected enzyme. Don't call it that way, okay? There, I removed the ass enzyme. It's no longer present, okay? I thought it was too disrespectful to an enzyme, right? Anyway, listen. I'm going to remove all this, but before I do, I just want to tell you something. This compound, this structure here, which is almost identical to this, is going to be called a nicotinic acid adenine dinucleotide. Okay, I just want you to remember this. Why is it instead of course, nicotinamide adenine dinucleotide, which is this, right? So what we're going to do here is that we're going to use the AS enzyme, or the enzyme, to generate NAD, right? Now, so I'm going to remove remember, the AMP portion, let's remove it, right? Because basically, we have only one step left, right? Let's remove the phosphate bridge, right? This. And let's remove the ribose sugar, okay? Good. So, the only step that is left is to take this carbon here, and the hydroxyl group attached to it, and put an amino group on it, which is going to be called an amidation reaction. Listen, we have an enzyme, a well-respected enzyme, okay? And by the way, listen, where do we get the amino group from? This NH2? Well, it's not just floating around the cell. It has to come from somewhere, right? It comes from glutamine. That's where it comes from, right? So glutamine is like it donates an amino group, right? And the way it donates it is in the ammonia form, NH3. Basically, ammonia is a nitrogen that's attached to 3 hydrogens, right? Then we remove a hydrogen group, and so we're left with, well, an amino group. So how exactly can we do that? Now remember, ammonia can't just be running around freely in the cell. It's extremely toxic. So this enzyme, it has 2 pockets, 2 active pockets. It is the NAD, because it's going to make synthesis of NAD, so it's NAD synthase, right? It has 2 pockets. One pocket is going to grab onto glutamine, right? And the other pocket is going to have, well, almost this structure here, but, you know, instead of NH2, it's going to have a hydroxyl group, right? And so this structure here, all of it, is going to be called the nicotinic acid, right? Adenine dinucleotide. And of course, it's going to require ATP. Why? It's not going to use ATP, it just uses it as energy. So the ATP is not going to be incorporated anywhere. The glutamine is going to undergo hydrolysis. It's going to give us ammonia. Remember, this is all occurring in this respected enzyme, the NAD synthase, or synthetase enzyme, right? Now, it's not going to go out into the cell freely. Instead, there's like a channel here. So the NH3, it goes here. The glutamine is reduced to glutamate, right? Okay. And the NH3 attaches itself to this ring right here. So the hydroxyl group is now gone, and we have an NH2 plus a hydrogen, right? Because it's not going to be ammonia. So basically the reaction would go something like this. Listen, the nucleic acid, adenine, dinucleotide, plus glutamine, right? Plus glutamine, which gives us the NH3. Plus ATP, because we do need energy for this, is going to yield what? What is the product, the end result? Is going to give us NAD, this, plus glutamate, because now remember, when we take an amino group out of glutamine, we get glutamate, plus AMP, because we use 2 phosphates here, which are here, plus the pyrophosphate. This is not important. I just want to show you how an enzyme, different enzymes have different active pockets and even have different numbers of active pockets. So something like a hexose enzyme has only 1 active pocket. This NAD synthase had 2, right? Some enzymes like the hydrogenase complex, right? It has 3. So active sites, people. Okay, I just wanted to show you the reaction that occurs. Anyway, eventually, when we get, but the glutamine, by the way, it gives us the NH2, remember, and we put it here. So now, instead of a hydroxyl group here, what do we have? What do we get? Well, we're going to get an amino group. Now, because this amino group is attached to a carbon, and this carbon is attached to an oxygen, right? it's going to be called not an amino group, but an amide group. And so this process that we just talked about is going to be called amidation reaction. All right? And that's the thing. This is how we took a nicotine and really eventually formed a NAD+, right? Now, that's the Preiss handler pathway. What about the salvage pathway? Salvage pathway, listen, in the salvage pathway, you already start with a nicotinamide, which is this. So the only thing you have to do, it's really a shorter pathway. The only thing you have to do is, you take the phosphoribosyl pyrophosphate and you stick it onto it, right? And then you take an AMP and then you stick it onto it. So basically, the first two steps, right? Where the enzyme is going to be, exactly the first two steps as in the Preiss handler pathway, which we already talked about, right? But the amidation step in the salvage pathway, we do not do that. That's all it is. The last one is the tryptophan pathway or the de novo synthesis pathway. This we're going to talk about in the third lecture, right? The clinical tie-ins. You want me to show you? Listen, I'm going to do it. If you want to see how the process occurs, continue. If you do not, just bye-bye, okay? Oh, you know what? Oh, okay, let's do this very fast. So, over here, you have a nicotinamide. Now, this is a nicotinamide, right? Nicotinamide, why? Because it has an amide group. So, instead of nicotinic acid, it's a nicotinamide, right? Great. So, now what? Now what? Well, you take this phosphoribosyl pyrophosphate, right? Here's a phosphate on the 5', and on the 1', there's two phosphates, and we're going to link it. The two phosphates are going to go bye-bye, right? And basically, we are going to link like this. Now, this is called nicotinamide mononucleotide, right? So here, nicotinamide mononucleotide. Okay, that's good, great. Now, well, what else do we do? Well, what else you do is you take the ATP, and remember, here's the ATP. ATP is adenosine monophosphate. Here's the adenine, and here's the phosphate, ATP, right? And you take the adenyl portion, which is going to be this, which is simply the AMP. We call it adenyl again. And through the enzyme, this time it's a kind of different enzyme because, it's a nicotinamide mononucleotide adenyl transferase, right? And you transfer this portion, and basically there's an oxygen bond that's going to occur between them, and we're done with the salvage pathway, right? It's for completeness sake. Of course, these two are going to go, right? Here, it's the salvage pathway. It's much shorter, and maybe it's shorter because we talked about it already, right? And you don't have to do amidation because, the amide group is already there. This was the biosynthesis of NAD. This is how NAD is made. So basically, again, NAD, 3 sources, 2 of them from vitamin B3. If you're eating meat, if you're eating liver, if you're eating eggs, you're going to get nicotinamide, right? And even though it is not really recycled because you took it from the dietary place, right? Or dietary supplement, or took it from the diet, it's still going to enter into the salvage pathway. So nicotinamide can come in 2 ways. It can come from the diet as nicotinamide, or it can be present already in the body after we use the NAD+. Remember, it's going to get dissociated and destroyed, and then we recycle it. So this is called the salvage pathway. Or you can get nicotinic acid, usually from like legumes, corn, plants. Of course, the bioavailability is not very good, and I'm going to tell you why in the third lecture. And number 3, de novo synthesis, right? Something that has to do, that really, it makes the end result of it is NAD, okay? Very long pathway, not important at all. So see you in lecture 3, and where we're going to talk about the clinical significance. 3 is like a 10 star, if I could, rate it. Have a good day.